Good evening. My name's Helen Reeves, and I'm sitting here with Tom Luedde.
Hi.
And we're here just to tell you a little bit about the EASL HCC summit that's been going on in Geneva over the last couple of days. And this is a meeting that is the second one of its kind, organized by EASL in a scientific committee and the EASL office. And it's really been to focus both basic scientists and physicians of all different specialities together in the same place to really move forward the field of both managing patients with liver cancer and the research that's relevant to progressing the field and teaching our juniors and young investigators and wanting to inspire them to actually learn more about liver cancer and be involved. And all of the sessions that we filmed here at this meeting will be available on the LiverTree, so for people that are interested in going to have a look at something that we mention in our discussions, please do that. Now, Tom and I, we were responsible for putting together the basic science part of the meeting, which has taken place over 1.5 days, and we decided that we would organize this around the hallmarks of cancer, and these are the features that define a cancer cell as described originally in a landmark paper published in 2000 in the journal Cell by Douglas Hanahan as the first author. And what we've done is invite people from our own field and a little bit outside our own field to get together to look at some of these features of cancer. And for our keynote lecture, which was one of the highlights of the meeting, we did actually have Douglas Hanahan here to talk to us about his own perspective of how they came to perceive these features or hallmarks of cancer. And what was very interesting from our point of view is that he helped to set that in the context of liver cancer, to give us some pointers to how he thought we should be looking at our disease. And he also gave us some insight into the way we ought to perhaps be approaching treatment for all cancers in the future. The fields tend to have focused on perhaps trying to target one of these hallmarks, whether it's proliferation or evasion of apoptosis. And what he described was this switching of the hallmark phenotype, so that perhaps as we move forward trying to tackle cancers, we should be thinking about a stepwise approach where we perhaps target proliferation, but before the cancer has developed resistance to that strategy, we should be thinking about the next one and how to implement it. So I think for both of us that was a very exciting part of the meeting.
Definitely. So Helen, this was, I would like to say, this was a great idea of yours to first put like the structure to this meeting, call it the hallmarks of cancer. I think in liver cancer we have had like times where we had different many systemic trials of systemic treatment recently had failed in the last years. So really we learned from this, I think, that we would have to think about new strategies. So the idea of the basic meeting was really to show the whole spectrum of the different kind of research that people in the field are working on now, and like giving it the structure of the hallmarks of cancer really opened the whole panel of potential ways how to tackle cancer. I fully agree with you that the keynote lecture of Douglas Hanahan was really like amazing, because he is a real vision. He's had really great visions. He's not working on liver cancer himself. He shared some of his own original work with us too, but really please, and I really advise everybody of you to watch this if you can, because as you said before, like the idea not to just target one target in cancer, but really think about combined targeting or sequential targeting of different features of a tumor. As you said, like targeting cell death evasion, targeting proliferation, metastasis, but also inflammation. This is something that is now really starting, you know, and I think the crucial point that we discussed afterwards would be if we can even define biomarkers that would tell us when to switch the therapy. And this takes me to the very first introductory lesson of Peter Schirmacher, who really started up the whole meeting. And I think we had a good, it was a good idea to put him first, because he was discussing, for me, I think you agree, one of the most crucial questions, how we can link the basic science knowledge to the clinical applications. And I really liked also your discussion that you started when you said, okay, and yesterday, or the first 2 days, really many people agreed, basically, we would need more biopsies, right? Really, where we would have to get more material, right?
Well, Peter Schirmacher is a pathologist, and he was actually also one of the organizers of the first HCC summit, and we knew that he would be a good person to bring back, just to bridge us back to that meeting as well. But as a pathologist, he's particularly keen on having biopsies, and we all are keen on having biopsies because we know that that's the way forward to identify the targets and to help to stratify treatments. But we did discuss that in the current guidelines, biopsy is not routine, and that's because, as we all know, that the sensitivity and specificity of radiological imaging in patients with cirrhosis is actually very good. It's how you balance the risk of a biopsy, or not, even if the risk is small, the procedure that the patient has to go through if it's not going to change the management of the person in front of you. So there's very much two camps: the people that think we're not going to move forward unless we do this routinely, and then the perhaps more balanced approach of saying, yes, we should do this, but it has to be part of a structured research program.
Exactly.
So that you're collecting the biopsies properly, biobanking the samples, collecting the data, and working together. And certainly that was Peter's, one of his main messages, really, was biobanking and tissue and those kinds of studies. We also then had, as part of our first session, an introduction really to some of the key ways of how to do these kinds of experiments that we all plan for the translation. And Jessica Zuckman-Rossi gave a very nice overview of the sort of genetic approach to studying cancer, how to identify drivers of cancer. She focused on TERT or mutations in the promoter region of TERT, and she helped define how that was a driver, or how she came to that conclusion. The disappointing fact was that it's not something we can target particularly well in cancer, and so these are all challenges that everybody recognizes. And then we also had some other very good lectures in that session.
Definitely, we had Robert Eva, and we gave him a very difficult task, I think, but we wanted really, because it was this basic science meeting and many people do great work, but we learned over the time that the models, it was a matter of discussion at many talks, which are the best models, which are the proper models that we can really work on, so our work is not just going nowhere, but it needs to go somewhere to the human situation. So Robert spent a lot of efforts, and it was really difficult. He couldn't show so much of his own work, but but I recommend everybody to see his talk if you can, because he spent a lot of efforts. He showed nice slides where he showed, okay, these which features of which models and so on. I think he should just
with
go
animal models.
Yeah, he should really go now write a review article. So to sum this up, because I think it's really urgently needed. And also we had a very nice talk of somebody who's not really in the HCC field. That is Twan Lovers, a very energetic talk, I think, and he showed some excellent models. He really spent a lot of time thinking what could be interesting for HCC research. He showed how he can even image biomarkers. Did you remember this?
Nanoparticles he was talking about, wasn't he?
Or these nanobodies that he showed from breast cancer, where people can stain HER2, it's called, right? So I was thinking we should really think about how we can maybe image biomarkers in liver cancer research. I think he's really, I hope to see him again at these meetings, and so he stays also enthusiastic about liver cancer.
No, I agree. And then we moved on to actually some of the sessions focusing on the hallmarks themselves. And the first one was on sustained proliferation, or methods of evasion of cell growth. And we had Erwin Wagner, David Moore, and Massimo Livero speaking in that session. And they're all very good lectures. I think the highlight for me was David Moore's lecture on CAR and beta-catenin. And the role that plays in the development of hepatocellular cancer. And what was quite fascinating really was that he took this back to sort of basic biology and talked about circadian rhythms and sleep and jet lag and how all these things affect us. And he actually showed very interestingly that sleep deprivation, either in mice or presumably translating to humans who don't sleep well or have sleep apnea, these things are associated with the development of fatty liver disease, steatohepatitis, and cancer. And so the take-home message for me was that as long as I got enough sleep, I would be slim and beautiful and that I wouldn't get cancer. But it was a bit more scientific than that. He did relate it back to CAR and beta-catenin signaling, so that was exciting.
This was really an excellent talk, yeah. But also the others were really amazing. Then, in the next session, something that is one of probably the hottest topics right now in
this is Tom's topic,
cancer.
So he thinks it's the hottest topic. I think I probably agree.
I mean, like in all parts of oncology, basically, the immune therapies really have opened a new era. And led to amazing results already in, unfortunately, only subgroups of small patients. So the whole question is, how can we get also these new therapies available or work in our patients with HCC?
How can we understand which cell type is responsible for causing a particular effect and how we can target that? And actually, it's not just the individual cell types, it's the microenvironment, including the stromal cells, and then the T cells and perhaps the neutrophils, and all of these things causing different things. And we had a very good little panel of speakers for this didn't we?
I mean, we had Derek Mann from your institute, from Newcastle. I think you're very active right now. I can say this, you cannot say it yourself, but I think that is a very active research group. Derek is really doing pioneer work on granulocytes and liver cancer. Then Robert Timme from Freiburg in Germany. He's really, he has been working on immune responses and viral hepatitis for years, and now he's successfully also worked on these kind of antigen responses in liver cancer. I think what I liked about his talk especially is that it was a very careful consideration of all the challenges of this infection. You know, why progress is not made like from until tomorrow, but it really takes small steps and also obstacles to overcome. And the way he really showed the difficulties that we're facing right now with antigen presentation, tumor antigens, how difficult these are really to show them even. And also then afterwards Tim Greten came and showed his, Tim Greten is a very successful researcher.
Yes, he's a leader in the field at the moment, isn't he? He's got some very good papers this year, as well as the talk that he gave.
Yeah, he published in Nature and Cancer Cell recently. And he showed his way. I think he found a very interesting connection between how the metabolic alterations in NASH influence immune responses, how they influence T cell activation and so on. And I mean also what I really found impressive, I saw his kind of study that he showed, and he just published it, I think, like how he can combine local ablative therapy with immune therapy. And he showed it in a very small collective of patients already, that when he's treating like one lesion, and a cancer lesion in the liver, and then he combines it with immune therapy, how then the other lesions really respond to this therapy. I mean, that was really amazing, I thought, and of course it was a very small cohort of patients, but I think that it's really...
Yes, I think there's this idea that you release the tumor antigen with one modality of a local regional therapy, and then you enable the immune system to see the tumor and then augment its response. So no, it's exciting.
And we had a great discussion, I think, in this session, really. So it's really worth watching it.
And actually, the next session we went on to is targeting angiogenesis. And here we had invasion and metastases, and we had some nice talks from Erica Vila, a more clinically based talk on the development of a 5-gene signature. This is something requiring biopsy, so going back really to this core theme. And then we had a fantastic talk on endothelial cells and angiogenesis, didn't we?
Yeah, that was given by Peter Camillier, also somebody who's not really like regularly seen at HCC meetings, but he's, I think, probably worldwide one of the most successful scientists, and he's he introduced to us his concept. So this is one thing that I remembered well, was this concept of vessel normalization. Yes. You know, as a new concept, how we should, so not the classic anti-angiogenic therapy, but you know, like to...
No, I mean, he started out explaining how we've tried to target angiogenesis and then failed, because something like the VEGF receptor signaling pathway, if you block it, it will only work for a short time, and then something like fibroblast growth factor receptor instead will stimulate angiogenesis. So he'd come up with the idea that you actually needed to target the endothelial cell itself, and because that cell's ATP production is dependent on glycolysis, it was a way to actually target that specifically, so to target that pathway, and then having taken us through the way he'd done that, to then finish up with, actually we don't need to stop the blood vessel formation, we need to promote it so that you deliver your chemotherapies better. It was fascinating.
I want these videos for my work. He did these fantastic videos. I hope people have a chance to see them. We had the next session was enabling of replicative mortality, genomic instability and mutation, and we had some very interesting talks in this one. Then we had one of the highlights was also we had Li Su, who is the editor-in-chief of Cancer Cell. We wanted to give like the meeting a bit also an educational aspect, and I'm very happy that he accepted our invitation, and he gave an interesting talk. We thought, okay, if he's coming, first of all, of course, we had the chance. I found this very nice chance to introduce our community, to give like kind of get in contact to also the editors. He's the editor of one of our most important journals anyway in cancer research. Show that liver cancer research is really important. What are the most important questions that we're addressing? And of course, to hear what he thinks. He advises us to make our papers a cancer cell paper. Of course, this was a little bit like provocative title, but really like to have the editor's view on what like also for the young scientists. I think it was a great chance now to see what really matters. You spend so much work on your
and for some of our senior scientists to stand up and tell him that he needs to make his journal more translational.
Yeah, but I think also he's doing a very good job to give it a space also for the molecular work. But definitely that was his message also. He wants the work, he wants the papers that really matter in therapy of human patients. Then
then we moved on to, a session we called Deregulation of Cellular Energetics, which is a very fancy title. It was more to do with metabolism, really, wasn't it?
Yeah.
And we did have some excellent lectures there as well. One from Matthias Heikenwalder.
Yeah. So Matthias is one of the rising stars for liver cancer research. He's right now has a position at the German Cancer Research Center in Heidelberg, and he shared with us his data, which I found really interesting because they're so translationally have so much potential. He showed us that inhibition of platelets, he showed data with aspirin, for example, really can improve like NASH and NASH-associated cancer. He really established one of the most important mouse models in NASH research, what I think right now, and I think he showed really that if you inhibit platelets, you can really improve NASH. You know?
No, I thought it was, I did wonder after his lecture, there's been so many patients in all these cardiology trials and stroke prevention trials treated with aspirin that we ought to be able to work out whether or not the patients who've been treated with aspirin have a lower incidence of hepatocellular cancer. But no, it was very good.
But I mean, we should state that he said that you should target platelets even more specifically, right, than aspirin, and he showed all these knockout stories. So we definitely will hear from this paper. Herbert Tilk from Innsbruck is a real expert, I think, in something that's also really like hot right now, the gut microbiome and what it does in liver disease, in NASH and liver cancer. I think he gave an excellent overview. So really, if you have a chance to see that online, his talk, he's a real expert, and I think he managed really to be in time, to the point, showed also his own very interesting data that he published now on colon cancer last year on the microbiome. So this is really something that is very important. Also in the same session we had Peter Janssen.
Yes, that was outstanding.
Yes, so Peter Janssen is also somebody who's not regularly showing his science at HCC meetings, at liver cancer meetings. He's an expert. He did really seminal work on cholestatic liver disease. That is really, he's really leading in that, and he showed us data on the topic that is very hot right now. You know, the new drugs.
Bile acids, yes, and FXR agonists, and people wanting to use these agents to treat whether it's primary biliary cirrhosis or non-alcoholic fatty liver disease. And so yes, he showed us some of his data.
Yeah, he showed us data that you, like in many cases in like when you have new concepts, there's two sides of it, right? So he showed us the beneficial effects and the molecular underpinnings, how these drugs work, how they improve like the whole system, how it improves the biliary situation and also the metabolic situation, you know,
yes, but he raised this concern that we need to understand, because there's a possibility that if you upregulate FGF signaling, that you might actually increase the risk of getting cancer, or perhaps promoting cancer progression.
I think his statement was very careful. I like that too, because he said probably it's a dose effect, because the findings were shown in transgenics who very strongly express FGF19, and probably, but maybe in the long term, that was his message that I really shared. We have to really just be very, very cautious, you know, and see if we might look for the signals, you know, from the big trials and so on, and from the data.
And then the last session was on cell death. That's your thing. Yes. There was an outstanding lecture by someone called Tom.
Thanks a lot. And,
it was very good. A lot of us understand the concept of apoptosis as a regulated means of cell death, and necrosis as being something that's not regulated. And you talked about necroptosis as something that's a regulated necrosis. And you've come up with a target.
Exactly. Yeah. I mean, this is a pathway that we're working on, and I really like to have this session with Robert Schwabe, to have the privilege to put the program like that, because Robert is like one of the most influential researchers over the years now in liver cancer research. You know, many people referred to his work at this meeting, and he shared some of his data on cell death responses, something that interests me also a lot. And we had a great discussion after his work, and this was then followed by the keynote lecture, as you mentioned.
Well, Douglas Hanahan asked you a question.
Yeah, I was really honored. It was a very interesting question, and I'm glad I read his paper again before I came.
I was glad you read his paper. But it was actually a fabulous session. We've had a great time. I think the juniors that have attended the meeting have also had an opportunity to present e-posters, so they're all available for people to look at. So it's been very interactive and very exciting, and hopefully there's all this history, the content that will be available on the LiverTree for people that have the time.
It was a great pleasure to do this with you, Helen, and also we should acknowledge the work of the people from EASL who did a great job, and I hope we can meet again at the next HCC Summit.
That's right.
I hope before, but it will happen.
Thank you.
Thank you.